in which we're going to be responding to Genetic Beast. First of all, I have to clarify that this is not the part 2 of the series that I was uploading into YouTube. Debunking what he says, this is like a intermediates between the first core of my critique and the second core that I am completing right now. Because this second core right here is incredibly long, and I still have to summarize a lot of articles and debunk some things that this guy has said. So since this is incredibly long, and I still have to do all of this and summarize all of this, I'm going to make a video while I'm doing that, like an intermediate between the first core and the second core, debunking the things that he's saying about mechanical tension. Which is that mechanical tension is not the thing that is causing satellite cell activation. He mentions that in these two videos, and we will... Oh, sorry. And we will, also analyze his article that he presented right here in this video, which I have right here. And, well, let's start with the critique. First of all, we have to know what his argument is. He's saying that muscle tension is not the thing that is causing satellite cell activation. The thing that is causing the satellite cell activation is damage caused by muscle tension, because damage causes satellite cell activation. So he's saying that muscle tension alone, which is a factor, is not the thing that is causing satellite cell activation. The damage that is being produced is mediating this activation. Well, this is simply wrong, because muscle tension can be the same when damage is being reduced, because we can reduce the load that we're using, or we can reduce the effect that is causing on our fibers. Right? We can use the protective effects of the repeated bounce effect, which we have mentioned in the previous video, and we have proven why it is necessary for you to grow. And we have also proven that muscle tension is not proportional to, the damage is not proportional to the amount of myofibers growth that you receive. Well, we have disproven a lot of things that he said in the last video. But the main thing that is the topic of this video is mechanical tension, right? Well, first of all, we have proven that muscle tension is not proportional to damage because of these two things, which are the repeated bout effect and small loads. So, considering that muscle tension is not proportional to damage, we cannot say that damage is the thing mediating satellite cell activation, unless the amount of satellite cell activation that is received when the amount of damage caused by muscle tension is less. Let's say that muscle tension happens and we receive less damage. Satellite cell activation, if damage was the factor that was mediating this, should be less. Well, we have proven in the last video that the amount of satellite cell activation by IGF-1 signaling and another factors and interleukin-4, which is another factor right here, it doesn't change at all, because muscle tension still leads to the activation of these kind of factors. But this is not the thing, or this is not the way in which I want to debunk all of the things he's saying, because he would try to debunk all of these mechanisms by which muscle tension is being felt by your fibers, and in response to that, some kind of activation of satellite cells and proliferation and all of that happens, saying that the thing that is eliciting these mechanisms is the damage, even if it is small. That is the thing that is causing these mechanisms to activate. So he would say, like, for example, that sarcopenic mechanosensors work by the damage that is being produced by muscle tension, right? He would say something like that. Or that mechanosensitive substances, like interleukin-4, are not sensing damage, but they are not sensing muscle tension, but are sensing damage, like right here, which says that it has been identified as a mechanosensitive cascade and all of that. Well, let's take this sentence, for example, that will debunk all of this argument, which says, it is conceivable that when muscle fibers sense tension, they communicate via various factors to recruit satellite cells and contribute minuclear to support hypertrophy and potentially stabilize the minuclear domain. The first point is that it is not talking about muscle damage at any point in this sentence. Any point that this situation that they are describing implies damage. So damage is something that they are not mentioning. This citation right here and this description of interleukin-4's behavior is not saying at any point that these mechanisms require also damage to be activated. These sentences right here are only describing a function for these mechanisms that they are talking about when it comes to fibers sensing tension. So you are making an unwarranted assumption when you introduce damage to these mechanisms, because it doesn't play a role in any of those. you have to justify why these mechanisms right here that they are describing require muscle damage to work, since they are not assigning any role to muscle damage in their mechanisms, which means that they are making them independent of muscle damage. They are literally saying that these mechanisms are independent of muscle damage, and you're saying that they are dependent, and you are not providing any kind of argument to justify your position. So that's unwarranted assumption right here. Unwarranted assumptions are claims or beliefs that possess little to no supporting evidence. That's what you're doing right there. And well, I am here again. I had to something, but why I'm here again right now. So let's keep debunking what he's saying. Well, we have just debunked his claim using one counterargument, but there are multiple counterarguments to debunk what he's saying in most of these videos that we will review after explain all of these counterarguments so that I don't have to repeat them every time we're watching these videos, okay? So, well, let's go with the second counterargument to his claims. First of all, let's take this based upon that sentence, the only requirement established for effect to happen is sensing tension, right? Nothing is said about damage. Therefore, and also damage is not a direct consequence of mechanical tension. So if it's not a direct consequence of mechanical tension, then this tension that is being established as a requirement is not always being accompanied by damage. Therefore, this lever that activates this mechanism is not always activated at the same time in which damage of some sort is being produced in the muscle fibers, right? So, based upon that sentence, the only requirement established for the effects to happen is sensing tension. Yeah. Therefore, if myofibers sense tension and there is little damage, or if myofibers sense tension and there is a lot of damage, the same effect will happen, because the myofiber sensing tension is not going to be always accompanied by a lot of damage. So, if this mechanism is directly affected by muscle fibers sensing tension, and in not every occasion in which muscle fibers sense tension, there is a proportional amount of damage. Let's say muscle fibers sense tension level 1, and recruitment of satellite cells is in level 1. The amount of damage in each one of these situations that we have generated, yes, will be different. Therefore, if damage is bearing because it is not proportional to muscle tension, and satellite cells at recruitment is still proportional to muscle fibers tension, as described by this mechanism right here, which is taken from this sentence right here and from what Max is saying, then damage cannot be a regulator of this mechanism since it is always bearing, even though the mechanism is producing the same effects. So that debunks what you're saying. Let's keep reading what I said here, because it is the same that I have already said, but it will be quite good if we expanded upon that topic. So, the requirement established for effect to happen is sensing tension. Therefore, if my fiber sense tension, there is little damage, or if my fiber sense tension, there is a lot of damage, because there will be a lot of occasions that if we generate based upon this mechanism will produce exactly the same effect. Because suppose that we simulate these situations using this mechanism that we have proven. Well, in each of those situations in which damage is varying, the same effect will be produced based upon this mechanism that we have described. So damage simply is not the thing that is regulating the mechanism. So your claim is wrong. Muscle fibers tension is not regulating this effect by damage. Based upon this mechanism that has been extracted from this sentence right here and has been proven in this article right here, and not only is proven by that article right here, but also is proven by action of multiple molecules that respond to tension alone, like interleukin-4, and also sarcomeric mechanosensors. Mechanosensor, what is a sensor of, what is a mechanosensor? It's a sensor that detects when tension is being exerted, okay? So it is actively watching for tension happening, right? Since its task is to watch for tension and see if tension is being exerted upon the muscle or not. Since it is only watching that, as it is his only function as a sarcomeric mechanosensor, then it doesn't care at all about mechanical strain alone. Just a mechanical strain provokes these proteins that are present right there to change its attitude, to change how they work. And since tension is activating these mechanisms alone, and the damage from the other fibers is really not activating this mechanism, or at least there is no mechanism that explains how the damage that is being suffered by these myofibers right here. Let's say that you're doing some curls, bicep curls, and you receive some damage. There is no mechanism that explains why that this mechanism right here has only been proven to work using activate muscle satellite cells. There is no mechanism that we can use to link damage to muscle lean protein. There is only a mechanism that we can link muscle tension to these proteins activation. So we can only explain it by tension of the fibers, and we cannot explain it by damage. That's, in short, what happens here. We have another one, anchored repeat domain under muscle anchoring repeat proteins, half-link domain proteins implicating cardiac hypertrophy, or the M-band where the kinase domain of titin may act as a sensor of mechanical signals. Yes, mechanical signals. So you're since you're experiencing some kind of mechanical loading in your muscle, and also you have some kind of tension, and there is a mechanism by which that tension activates all of these proteins right here. But there is no mechanism by which the damage, as a product of that tension, activates these same mechanisms. So we cannot say, since there is no mechanism that links damage to the activation of these proteins right here, there is no mechanism that can explain why damage should be the factor that is activating these things right here, and not tension, since it has been proven, these citations, the tension mechanistically, yes, mechanistically activates these proteins. We cannot say that muscle damage is the thing controlling these mechanisms right here, because we have only proven tension to be a regulator of these mechanisms. There is no mechanism that links muscles, yes, muscle hypertrophy, myofibers growth, to damage in these mechanisms described right here. Yes, I said the word mechanism a lot of times, but that's the topic that we're talking about: mechanical tension, mechanical tensors, and all of these things related to that. Also, we have a lot of studies right here that prove that not only do you need the reproducible effect, we have reviewed that in the last video, but that you can receive some significant amount of growth, and the same amount of growth that other people using lithium to age, an energy mechanism, energy expenditure, and gender, right? Whoa, this study right here that I am talking about, the study that he tried to debunk by using this study, has, first of all, control for all of these things that correct as confounding factors for creatine kinase, provoking damage, being an indicator of damage in your muscle, which means that all of these things that can affect the DCK markers, like gender, age, and all of that things, they had them under control, because 40 healthy university students, 8 men and 6 females, were divided into 2 groups with a composition that equalized X, sex, age, height, body mass, and strengths of the quadriceps. So all of the people that participated in this study, or let's say the 2 groups that participated in this study, had a similar age, a similar height, similar body mass, and a similar sex. So those things that are being considered here as confounding factors for these indicators are being controlled completely, so that they do not disturb its activity as an indicator of damage. That's why it is called why these studies are called randomized control trials, so that these factors that can affect the ability of these indicators to tell you how much damage, for example, the subjects are experiencing, these factors that can confound the study are all being held under control and are all out of the table. So no one should care about them, since they are completely under control. All of these factors that could confound the result of the study. So all of these things that this study is saying that could affect the results of the study, or the results of the creatine kinase levels, without any damage, are being controlled for. All of these confounding factors are being controlled for. So they are not exerting their confounding effects on this study. So this does not debunk at all my first study. It completely fails to do so. And the other thing that it mentioned is that CK is really important in energy management, and it increases once energy management is increased. The thing is that, for example, eccentrically based exercise will elicit greater post-exercise levels of serum CK than equivalent concentrically based exercise, though the former is less energy metabolism demanding than the latter. So eccentric movements demand less energy than concentric movements. So we cannot say that energy expenditure is the thing that changed the levels of CK instead of the amount of damage received, since they did the exact same exercise and they expended the same amounts of energy. So all of these factors that this study mentioned that could be confounding for studies are being controlled in this study and are not causing any kind of trauma. So that is one thing that I wanted to debunk. Now let's go to his video. We have already debunked all of the things he said about damage being the things that activate sarcopenic mechanosensors, even though there is no mechanism by which damage can be connected with sarcopenic mechanosensor activation. And also we have proven that out of these two factors, damage and muscle tension, only muscle tension has been proven to activate sarcopenic mechanosensors. Damage has not been proven to activate sarcopenic mechanosensors, so he can't say that damage is the thing activating them, because it is an unwarranted claim, and we have also proven, well, it's all proven right here and in my last video, so I don't have to repeat all of it, right? Now, let's go to his video right here.
Alright, so we all know that muscles have to so that they grow bigger,
Yeah, that's true. And now he's saying it's not.
No, it's not the case, and well, of all of the studies that I have mentioned right here to prove it, mechanosensors, mechanosensitive substances, nitric oxide released during exercise, even if you are receiving damage or not, because there are mechanisms by which nitric oxide is linked with muscle tension, and in these mechanisms, or let's say for these mechanisms to work, you do not require any damage, or at least damage does not play a role in the correct functioning of these described mechanisms. Like, for example, where is it? Nitric oxide. Nitric oxide, where is it? Cannot found it. Wait, let's use search. Right here. Recent research provides evidence that factors released in response to mechanical tension. So, there is a mechanism by which tension alone produces the release of nitric oxide. And it does not involve damage, since it is not being mentioned right here as something that contributes to these events right here happening. If it doesn't mention damage playing any role, then you cannot say that damage is playing a role. That's simply interpreting evidence in such a way that it benefits your bias, or that it goes along with your bias. That's called confirmation of bias. Right here. Well, it's in Spanish, so let's use this site right here, which is in English. Is the tendency to search for, interpret, favor, and recall information in a way that confirms or supports one's prior beliefs or values. So you're doing exactly that. And you are not even proving what you're saying. And it is even explained why tension alone can release nitric oxide, because nitric oxide can be released from differentiating muscle cells in response to stretch. Stretch alone. You stretch your muscle cells. They can receive any amount of damage, but this damage is not the activator of the mechanism. It's not described to activate the mechanism. It hasn't been proven to activate the mechanism. The only thing proven right in this citation right here, which is inside the article, the 126, the only thing that was proven inside of that article to promote satellite cell fusion is by this mechanism right here, is muscle tension. Damage has not been proven to have a role in this thing right here, right? Or even if it can activate this mechanism, this thing right here activates independently of damage. So if there is still tension happening, this nitric oxide will be released independently of the amount of damage that is being suffered on the muscle fibers. So we cannot contribute the release of nitric oxide to this confounding factor, right? Because of this damage right here has not been proven to be a confounding factor in for muscle tension releasing nitric oxide, and also interleukin 4 and There's also a mechanism that I will talk about in the second core, that is right here. Muscle stretch or injury. It says muscle stretch or injury. It's separating the two things. If it wanted to talk about injuries induced by muscle stretch, it would only say injury right here, because injury already includes all of the damage that is being produced by muscle stretch. So if it's mentioning this thing apart from injury, it means that it is talking about muscle stretch alone. It's basic comprehension of text, so that is very easy to understand, right? You are very unable to understand basic texts as you have proven in all of your videos. Let's try to expand this right here. Let's keep up with the video.
Hey, muscle fibers so they can grow back again stronger. Let's see what he says. He's gonna contradict himself because he's an idiot. Watch him contradict himself in this video because he said he works for Gymshark. He's employed by them. He's a fake Maggie, trust me.
What? No evidence was provided for these claims whatsoever. And well, you can work for Gymshark, Gymshark, and that doesn't necessarily mean that all the things you say will favor the narrative of Gymshark. And also, what does Gymshark have to do with muscle tension causing myofibers hypertrophy? Because I really don't understand why you wrote that up.
Okay, so he says that's not the driver of growth, the microdamages to the muscle fibers. It is mechanical tension that is the driver.
No, the two things are the drivers of growth: mechanical tension and damage. Mechanical tension can elicit growth by two methods. First of all, it produces damage, which has been proven to produce myofibers growth. But also, it activates sarcomeric mechanosensors. Yeah? And also, it releases nitric oxide. This muscle tension makes it so that nitric oxide is released, and it causes another mechanisms to be activated. Well, it's all right here. We have already reviewed all of that. And when nitric oxide, and also it activates IGF-1 pathways, and it is mechanistically linked with that. So we can see every step of this process in which tension ends up producing a further growth by release of nitric oxides and activation of these mechanisms. So we know that since this muscle tension produced at the start of the situation is producing a cascade of effects in which each one this muscle tension is participating, it's a main factor. We therefore know that all of the effects produced by this mechanism are produced by mechanical tension and not by the possible confounding factor of damage, since it is described step by step. For example, in this nitric oxide part. Let's look for it. Nitric oxide. Recent, we're going to look it again so that it's clear, because I think that I haven't explained it very well. Recent research provides evidence that factors released in response to mechanical tension, muscle fibers, and their associated satellite cells. So muscle tensions make it so that these things are released. And it is not mentioning damage, so we have no reason to believe that damage is the thing that is causing these effects, such as hepatocyte growth factor and nitric oxide may facilitate satellite cell activation and proliferation. So it is possible that damage can also make it so that nitric oxide is released, but they are strictly talking about mechanical tension. If they were talking about damage, or if the damage produced by mechanical tension was the factor that activated this mechanism, by which is initiated by mechanical tension, if damage was the main factor that activated and promoted these events right here, yes. And also, muscle damage can be produced without significant tension, as concentric actions also produce damage, as we have proven in the video in the article of Schrammfeld that compared the concentrically exercised arm and the eccentrically exercised arm. Then they would encompass damage as the main cause of the activation of this exercise, since talking about mechanical tension only talks about one of the ways in which you can elicit the damage that ends up producing these two things right here. So, if damage was the main factor that contributed to this activation right here, this release of things right here, then they would only be talking about damage. Since talking about mechanical tension wouldn't consider the damage produced by actions that are not very hike in terms of load and tension being produced on the muscle, right? Like concentric actions. So since it's clarifying mechanical tension and not damage, we know that they are talking about mechanical tension as the activator of this mechanism.
The growth. Well, that's funny because if you type up mechanical tension, build muscle, well, Google will tell you when a muscle is subjected to high mechanical loads, that is, it is in resistance strain, the fibers experience something known as what? Microtrauma. What is
And that is false. Not in every situation in which you are subjected to high loads, their fibers experience microtrauma. The repeated load effect makes it so that you experience less microtrauma, and even though there is less microtrauma, the requirements for all of the mechanisms that were shown in this video to activate are still being met, because mechanical tension is still the same. And so the effect, the final effect, which is activation of muscle satellite cells, is being elicited in literally the same way, because these mechanisms operate independently of muscle damage, and therefore it is disproven that the only way in which muscle tension elicits myofibers growth is by damage. Easy to see why, right? So let's keep up with this.
There's microtrauma. It's muscle damage. This microtrauma triggers a series of events within the body.
Yes, it is saying that one of the ways, it is not saying that when a muscle is subjected, it is not saying that the only thing that is causing the myofibers growth, once muscle tension is being, mechanical tension is being produced. It is not saying that the only way in which mechanical tension contributes to myofibers growth is by damage. It is saying that it contributes to growth by damaging the muscle fibers. It is not saying that it only contributes to growth by damaging the muscle fibers. So that's basically not comprehending the text that you have directly in front of your face right now. So that's very lame, but okay.
Including inflammation, protein synthesis, which ultimately leads to an increase in what? In muscle size. What he says, that's dispelled. You can't get these microtraumas to build muscle. It's mechanical tension, but mechanical tension is microtrauma. I think
mechanical tension is not microtrauma, because you can have mechanical tension with less microtrauma, but still have literally the same amount of growth, because these mechanisms are not watching for the amount of trauma that you received. They are only watching for the amount of mechanical tension that you receive. And these mechanisms, in situations in which mechanical tension has happened and activated them, no one could find whether the damage associated with this mechanical tension was really the thing that activated the mechanism. It was only shown in these situations and in this research exactly that the tension that these chemicals that react to tension and the tension that the sarcopenic tensions experienced, that was shown as the only thing directly connected with a mechanism to the activations of these pathways. So damage is not a contributor, right, to these situations, because the only thing that was proven to have a mechanism by which it is connected to activation of all of these things is tension. The possible damage that was elicited with tension, that, as I always repeat, the amount of muscle tension that you receive is not proportional to the amount of damage that you receive, and therefore we cannot say that muscle tension is damage. Easy. The only factor that was proven to activate these mechanisms, that was linked to activation of mechanisms like this, while muscle tension was on and damage in some way, probably in a very little way, was also being produced on the muscles. The only thing of both of those things that was shown to activate these mechanisms was tension. Damage was not linked in that situations to produce this effect. And also, these things right here, these compounds are mechanosensitive, right? So if you take them out of the muscle and you elicit some kind of tension on that compounds, then they are going to react. So even in a situation in which there are no myofibers, they activate by tension. Easy to see why. And while not most of them can be activated by tension in situations in which there are no muscle fibers, but the environment in which they are is experiencing tension, like interleukin-4 and all of those, but just tension signals the cells to release that compounds. So the cell just acts in response to this damage that can be produced in other parts of the myofiber and says, well, let's release all of these compounds. Like, it opens the door. It's not the case that the door is being broken by an axe or something like the damage that you could experience via training. The cell is just experiencing this tension And it's opening the door so that these compounds go to the outside of the cell, the extracellular matrix, right? It's not the case that the toric is being broken, and that is the reason why these compounds are being leaked onto the extracellular matrix. The cell is literally open its membrane and making some, I don't remember the name, but they are called vesículas in the Spanish, so that the compounds that are being produced inside of it go to the outside of the cell, right? Easy.
People are so...
The door is not being, for those that didn't understand me, because I usually really expand on all of the points that I make, the door through which these compounds are leaking to the extracellular matrix is not being broken, is just being open. So damage is not the thing that is causing these compounds to leak to the extracellular matrix. It is just the cell that it's releasing that compounds at its own will.
delusional out there. Even if you type this in Google.
And also, it is quite funny that his source for all of the things he said is just a Google search for the first thing that appears right after that. We don't even know what page is this one, so it's kind of funny. And also, it's a nutrients page in the tree. I probably some kind of whey protein page or creatine or that produces some kind of supplements. So it's kind of funny that his only source for that is a supplements page, but, well, it's obvious that it doesn't have any credibility, right? But, well, it was funny to me.
You can't get these microtraumas to build muscle. It's mechanical tension, but mechanical tension is microtrauma. I think people are...
Mechanical tension is not microtrauma because the two things are not proportional one to another, so you can't say that. That's dumb. And you also know how the repeated out effect works, so why would you say that?
So delusional out there. Even if you type this in Google. Is microtrauma an injury? So now let's investigate where...
This is very, very stupid that he has to search if microtrauma refers to injuries. Trauma of any kind refers to injury. How could you not know that, even though you have all of these studies that you haven't even read because you only read the abstract? How do you have such lack of knowledge so that you have all of these studies but you don't even know what trauma is?
Microtrauma is microtrauma. Or tissue injury, because that's what it is. In muscles, can affect both the central nervous system, CNS, and the immune system. Activation of the CNS by cytokines provides...
So you're saying basically that in response to exercise, the main thing that is eliciting the strength that you receive is the central nervous system, not the androgen receptors, like you say. Because if you're saying that this tissue injury or muscle injury can elicit CNS activation or can affect the CNS, then you're basically saying that one of the things that regulates the action of motor units inside of your muscles is being activated by injuries. So you're saying that the main way by which you gain a strength, you're basically proving with that thing you brought up, one of the main things that control the strength, which is the central nervous system, is affected by micro-injuries. So you're proving that your gains in terms of strength come from CNS adaptations. So you're basically disproving the thing that you said about androgen receptors that I already disproven in my other video once. That's funny.
It's a way of explaining many of the signs and symptoms of the OTS. So he says that muscle damage is not the driver. What mechanical tension is. What mechanical tension is muscle damage.
Mechanical tension is not muscle damage. Even the definitions are different. Mechanical tension is the amount of force that a string, let's take a string, for example, a shoelace, experiences when both of its ends are experiencing some kind of force. So that lace or that string is tensioned, and well, it experiences tension. And muscle damage is just the rupture of muscle fibers, which can happen with ROS, can happen with inflammation, can happen with osmotic pressure. So there's a lot of things that cause damage that are not muscle fiber, that are not, sorry, I don't know why I even said that are not mechanical tension. So you cannot equate the two things when they're completely different, and one can happen without the other happening. So not only are they different, but one thing does not imply the other.
It's microtraumas. What the fuck is he talking about? So that builds up non-muscle damage. That is muscle damage. And microtrauma is muscle damage. Just another word for damage. You're damaging muscles. You're always damaging something in the gym. There's something where that could be hypoxia. I don't know if you're chronic liver hypoxia. I don't know, oxidative.
Hypoxia is an environment in which there is reduced blood flow. But, well, if he's talking about a hypoxia, then he's basically debunking the fact that muscle damage elicits growth. Because when you have hypoxia, the amount of damage that you receive by this method that I have talked about in the last video, which is the Katsu method of training, I don't know where it was. I believe right here, katsu, yes, which elicits less damage, still produces the same amount of hypertrophy, so you're basically proving yourself wrong with each thing that you say. It's funny.
Could be a lot of bullshit you're getting there. Maybe nothing. I don't know. That's what you're doing. If you're so adapted. If you're so adapted, you're not getting these micro injuries, even if you're
If you're not adapting to the exercise that you're experiencing, then all of your fibers, all of the fibers that you have, are getting considerably weaker, because since they are not receiving any stimuli, any constant stimuli, so that they get stronger and they resist that stimuli in a better way. Let's say, if they are not receiving this constant stimuli, then you are promoting the existence of weak muscle fibers inside of your muscles. So you're literally doing the opposite of what you want. You're getting weaker by training in this periodic lifting manner, which is not really period lifting, because in order for you to do period lifting, you should have to wait 8 to 12 weeks. Yes, for the repeated bout of it to wear off. So that's wrong.
arguing chemical tension. It's protected by repeated bout effect phenomenon. Not damage.
Is to stimulate
Yes, and you require the repeated bout effect to row, as we have proven in the last video.
your fibers is significantly stronger, not damage them for
What? So you want to send a signal to them to get stronger, but not damage them. Micro injure trauma them, injure them, whatever, damage them. To get bigger, stronger. You want to send a signal for them to get stronger. Wow. Okay, that's funny. But here's I think I know what talking about this stuff here.
He's trying to search for myocytic androgen receptors, which is quite funny because I have a study in the second cohort that I would not review right now, because it's not the topic of this video, that proves that myocytic androgen receptors control the limb mass in muscles of humans, because the study that he's going to use is based on rodents and has a lot of problems, because in that study we have a group of muscles that are being affected by the androgen receptors, and in terms of mass, and there are another group of muscles that are not being affected by the androgen receptors, and increasing their activation, which are the limb muscles, have the peroneal muscles, I believe. Well, it's not something for the other video, so I don't want to expand a lot of it, but you have these things that elicit that androgen receptors really do control the amount of mass that the muscle gets. It is just the case that in rodents it just controls the mass of the limbs, but there's a study in humans that shows that it does not only control the mass of the peroneal muscles in the case of humans, it also controls the mass of the limbs. That's another study apart from that. But the funny thing about this study about myocytic androgen receptors is that the authors claim that there are a lot of discrepancies between all of the studies that have been made about androgen receptors in rodents. Like, no study has the same result one from the other. And I would like to go through each of the difference in the studies that they mentioned in regards to their own study. But, well, that's something for the other video, right? I don't want to spoil all of it, obviously. But that's just wrong, what he's going to say here. And the study itself does not prove anything. Remember that one of the main pillars of the scientific method is the capability of replicating the experiment. If you cannot replicate the experiment, then it is not scientific proof. So if we have this amount of discrepancy between experiments, we don't have scientific proof. I don't even believe that he knows what the scientific method is, but that's okay. Huh? This is really funny because he doesn't know how to use the keyboard inside of his how can I say it, in his screen of the cell phone. He's like a 60-year-old man trying to know how this strange thing works, like this magic telephone. Let's see if he says something of relevance in the rest of his video.
How did he get bigger? Because it disappeared after the games. How is he able to get bigger? Nobody else is screaming about.
What is he saying?
So what exactly is he getting bigger in the gym?
Yes, an eccentric exercise also elicits lactic acid release, and that is also related to the activation of muscles, sadly cells, but that's something that it's already included in the second Cora also.
How did he get big? How did he get bigger? Because it disappeared after the games. How is he able to get bigger, but nobody else is all these other guys screaming about natty, whatever.
Because the repeated bout effect does not diminish the amount of gains that you receive. That's a false claim. And the repeated bout effect is needed for you to get stronger. Right here, you have the article. No, this is not the one. This one. Now, oh, sorry. It was here, I suppose. Generation of the muscle fibers. Now, where is it? Ah, yeah, this one right here. This article. We have an article here that proves that MMH, which is myofibers, muscular hypertrophy, can only start happening after a repeated bout effect has had its effect on individual. Yeah, see?
Limits, genetic limits, arguments, all this weird shit that everybody else is on steroids. How is he able for 1 week to get that big? He was a skinny kid, he was He was a fucking man. He was like an anorexic little
because he didn't train like you did.
kid. And he's only been lifting for 4 years. Go look at his original thing. How did he get to be like that? You gotta question everything. He was 17. He's like 20 now. In the last 4 years during COVID, that's when he started. I think 2019 or some shit. So he's been lifting for the last 4 years. You're telling me he's not like that for years. Man, I know so many people they can't get even close to even that. Look how much he's lifting here. 405. He's deadlifting.
A deadlift of 4 or 5 is not that impressive. I myself, in my first year of lifting, I could lift 160 kilos, which is 20 kilograms less than what he's lifting right there. And I am not consuming any kind of steroid. I believe that I can lift more in deadlifts because I have some kind of advantage when it comes to my body, because I cannot lift a lot in bench press. Like, for example, in the first year I started lifting, I could only lift 80 kilograms on bench press, which I don't know how much pounds it is, but I don't know why Americans use these weird systems like inches, foots, and pounds. Couldn't they just use the same thing that everyone in the rest of the world uses? Well, I won't criticize that, but let's keep watching the video.
405. You think this guy's... this is... I don't know. Whatever. Yes, you want to minimize muscle damage. You don't control the body. You don't minimize muscle damage. You want to know what's...
You can minimize the muscle damage by training more often so that the repeated bout effect has an effect on your body. You can...
Stops muscle damage, repeated bout effect. With repeated bouts, there's no...
Well, you can control the repeated bout effect because you can control the bouts of exercise that you do. So you're contracting yourself.
Muscle damage. It's an protective effect. And even when it gives you that gains in the beginning, that is the adaptation process. It gets stronger. These...
Well, if you lose the repeated bout effect, and the repeated bout effect is made so that your muscles are able to resist more tension and more damage, then you're getting weak muscles by ignoring the repeated bout effect. That's really what you want.
The structural damage.
Also, what is the purpose of stop training so that the repeated bout effect wears off? If while you stop training, even if you stop training for 1 week, you start losing the amount of myofibers that you get, and probably the growth that you have been experiencing for each week of paradise lifting, it's just the growth that you have lost from the last week. So
damage gets stronger, so we can't repeat any more damages to the muscle fibers. Do you understand me? Because it's resilient from getting busted again. It doesn't want to grow any more muscles.
You can still produce damage on your myofibers, as I have proven in the last video, because the repeated bout effect does not make it so that the myofibers stop receiving damage, right?
So whether you think you can control something or not, stupid.
Well, you're more stupid because you only read the abstract of papers and you end up doing videos like this in which you embarrass yourself completely. So I wouldn't say that he's stupid. I would say that the one who is trying to criticize him is stupid.
He's dumb. Yeah, it's extremely hard. Just force him and everything. Force him. Yeah, that's the way to do it. In an adaptive state, as a chronic lifter, endurance athlete.
If you're not adapt yourself and you go to the gym, then you're keeping yourself weak. What is the purpose of that? Oh yeah, I thought that I was only recording one part of the screen.
Endurance lifter.
Because of this thing right here. That I don't know why it is right here.
He just said, it detects this kind of attention, and that's what allows it to grow. But it grows through what? Microtraumas. He just said...
Not microtrauma. The thing that is being detected is the muscle tension itself. These mechanisms are not detecting the microtrauma that is accompanying this muscle tension. And also, since muscle tension is not always accompanied by fibers damage, then these mechanisms, since you're saying that they are controlled by damage, they should decrease the amount of activation, or they should activate less if the amount of damage elicited by the muscle tension is less. But the mechanisms work in the exact same way in regards to... these mechanisms work in the exact same way in situations in which there are little to no damage, or in situations in which there is a lot of damage. Because these mechanisms don't use that damage. That is probably being produced as a byproduct of muscle tension, since you cannot avoid damage even if you have the repeated bout effect activated on you. These byproducts of muscle tension is not being linked in any way, shape, or form with the activation of these mechanisms, right? Because, as I already said, the only factor that has been proven in that situations in which you have damage as a byproduct because you cannot avoid it, and you have also muscle tension, the only thing of those both things that are happening at the same time in experiments in which they're trying to look for mechanosensors for substances that are sensitive to mechanical signaling, yes, of those two things, the only thing that was mechanistically linked, which means that it elicited a cascade of effects that ended up producing the final effect, which is myofibers protein accretion, right? On satellite cells accretion by myofibers. The only things of those two that has been proven to have a role in the mechanism that they were trying to study was muscle tension. Damage did not elicit any effect that collaborated in that mechanism happening. The only thing that was proven in those studies to elicit the final effect by a series of cascade effects was muscle tension. So we cannot assign the results of these studies to the damage. If there is no link with the damage produced as a byproduct of mechanical tension with the activation of the mechanism.
Don't damage your muscle fibers. Don't get these microtraumas. What the fuck, man? This guy just acts stupid.
Sorry if I keep sorry if I keep extending myself with things that I have already said, but I really want you to understand that he's wrong, and I want to explain my points so that you don't end up not understanding at all what I'm saying, and you end up following all of his advice that is completely unfounded. Let's go to the next video.
You could call this, I guess, a Q&A something, but I don't know. I'm going to answer something in the comment section. Somebody was asking me before. I forgot to add this in the blog video, this part too, but yeah, I'm going to just do it now really quick. So, you know, about muscles and this, whatever. Fusion and beyond satellite cell contribution to...
Yes, I have the article right here, and I have a part that I want to show you. Which is... Let's search for it. Right here. A challenge to understanding what mediates satellite cell fusion to adult muscle fibers with loading is that it is difficult to parse the effects of external cues from muscle fibers and or mononuclear cells versus the cell autonomous effects of mechanical loading directly on satellite cells. It is basically saying that it is very difficult to separate the things that are external to a cell to the things that are internal to a cell and are activating these mechanisms after tension is being provoked on these muscles. Muscle fibers can grow appreciably without myofibers aggression when hypertrophy is induced by genetic or pharmacological means in vivo. So, this is basically proving that the myofibers domain size can be surpassed by myofibers aggression, which means myofibers hypertrophy without sarcoplasmic hypertrophy. So this is disproving the MND size limit, finally. And it is said right here that this would suggest that mechanical loading is a key stimulus for satellite cell activity during growth. Some studies report that satellite cells on muscle fibers can be activated in response to stretch. So you just stretch the satellite cells and they activate. The damage that the myofibers are receiving do not have anything to do with this stress provoking the activation on the satellite cells, right? And it says that they are mechanosensitive and can act autonomously with contraction in vivo. Mechanical strain on myogenic progenitors, which are other kind of precursor cells to myofibers, and the myonuclei that are being incorporated into the fiber, yeah? Causes satellite cell proliferation, but inhibits differentiation and fusion into myotubes, because remember that mechanical strain, yes, is mainly used so that these myogenic progenitors end up being activated like satellite cells, let's say. All of the other things are being regulated by other substances that are also released with mechanical strain on the muscle fibers, not on these myogenic progenitors. These results suggest that mechanical tension directly on satellite cells in vivo certainly affects their behavior, but may not directly stimulate fusion per se, but the substances that the cells release in response to mechanical tension that the myofibers release does. So, satellite cells also increase in number in response to in vivo mechanotherapy. So they are mechanosensitive, and your satellite cells boom gets bigger, right? And collectively, this data suggests that the muscle fiber contraction tension so it's separating the muscle contraction or tension from the damaging components of load-induced hypertrophy. So it's separating the two things. Here it is only talking about muscle fiber contraction independently of damage, right? So it is saying that muscle fiber contraction or tension alone drive satellite cell fusion, right? So it's disproving what he says. And recent research provides evidence that factors released in response to mechanical tension. So, it is not the case that these cells are being damaged because they are producing mechanical tension on these cells in such a way so that they are not destroyed. They are not destroyed, and they end up releasing all of these intracellular contents to the extracellular matrix. Well, they elicit a muscular tension on these cells in such a way so that they don't end up being destroyed because they have to study these subjects, and the subjects being completely destroyed by the mechanical tension exerted upon them is not a good thing for the eye. So they exert enough muscular tension so these cells right here do not break. So these cells haven't break. They haven't received any kind of injury so that their insides led to the extracellular matrix. But even if they haven't received any damage, they released nitric oxide and hepatocyte growth factor. And it is said in this study right here, so it is debunking what you're saying. The same study that you brought up is debunking you. And, well, these cells are not being damaged, they are not being destroyed, and even though they are not being destroyed, they are releasing these factors right here, by their own will. So this completely disproves what you're saying, and I am using the article that you brought up, and it says may facilitate satellite cell activation and proliferation. So it is, if it facilitates proliferation, it is basically facilitating differentiation, because differentiation of satellite cells is a direct consequence of proliferation. Because once they proliferate, some factors that like MyoD, MyMy, F5, and all of those factors activate and end up causing differentiation. But this is something that will be covered in the second core. So
loading induced skeletal muscle adaptation. Listen to what they say, loading induced. Because it's like this Max Usada video, where Max Usada says that muscle damage is not the driver of...
Just because it says loading induced, it is not talking about the possible byproduct, because as we have repeated in exhaustion in this video, the amount of damage received is not proportional to the amount of tension received. So you cannot say that the two things are the same, or that they are one thing implies the other, because they are not proportional. You can receive more muscle tension and not this an equivalent amount of muscle damage. You can receive even less muscle damage. So you cannot say that when it says loading induced. It is referring to one of the possible byproducts of muscle tension. That, again, is confirmation of bias. And an unwarranted assumption.
Muscle hypertrophy, mechanical tension or mechanical loading is. Mechanical loading is muscle damage. Duh.
Now, we have disproven this survey, so I won't disprove it, Anan.
Yeah, man. So it's like myonuclei overload. Myonuclei overload. Well, fusion beyond satellite cell contribution to overload, loading induced skeletal muscle adaptations. Okay, so we scroll down here at the bottom. It's a short section. I left it out.
Yes, just because it talks about overload, it doesn't mean that it's talking about a byproduct of mechanical tension, which is damage. And even so, we're talking about it as a byproduct and not an effect, because it is not proportional to the muscle tension, which means that you cannot say that muscle tension is the same as damage, nor you can say that muscle tension causes damage, right? Because these two things can vary. So the first one, muscle tension, can vary independently of damage. So you can have more tension and damage remains the same, or it's even less. So there is no causal relationship between both, right? So you cannot say that they are the same, nor that one thing causes the other. You can only say that one thing causes the other in absence of confounding factors.
By accident, I should brought it up.
Or in absence of repeatable effect, right?
Whatever. So mechanical 2.2, mechanical loading. Mechanical loading is muscle damage. Physiological muscle damage and satellite cell...
Mechanical loading is not muscle damage. We have already proven. I won't go around the same points over and over again, but you already know what that's right.
Fusion to muscle fibers. So how satellite cells fuse to muscle fibers? Through what? Physiological what? Muscle damage. Fuck me, yeah, man.
Not really, and I've already proven in the last video that muscle satellite cells can fuse without damage, because there is nothing that damage produces so that satellite cells can fuse. There is no mechanistical link between the effects of damage and the fusion of satellite cells, in the sense that, well, damage activates satellite cells, but damage does not cause anything that is needed for satellite cells to fuse into myofibers. And also there are a lot of factors that control satellite cell fusion, proliferation, and differentiation, because once they have differentiated, they will fuse with the nearest myofiber that they find. Because once they are differentiated, they have the structures to do so, right? And there are factors that are released without damage, like IGF-1, as we have proven, with less damage, almost no damage. Yes, a little amount of damage, But even so, we have disproven the causal relationship between muscle tension and damage, so doesn't matter. And well, it is released. This factor controls differentiation, and since it controls differentiation, and it's being produced with almost known or incredibly less amount of muscle damage, even though the same amount of muscle satellite cells are being produced, it is basically proven, it's basically proving that even though you have no damage or little amount of damage, these satellite cells that are the same amount of satellite cells that you have elicited in the group which receive damage, well, not the same, because they didn't receive the same amount of hypertrophy, because we have a study right here that proves that you require the repeated bout effect to receive significant amounts of hypertrophy. But let's say that they have a similar amount of hypertrophy one to another. Not really. The group that received the repeated bout effect received considerably more hypertrophy in terms of myofibril growth, because of the study that we have shown right here, citation number 6, right? Well, IGF-1 is being released even though we changed the amount of damage, and the same amount or even a greater amount of satellite cells are being released even though we changed the amount of damage, and since the satellite cell pool has been completely activated and there is little damage, all of those extra satellite cells end up fusing with the myofibers, because once the asymmetrical division has happened, the satellite cells that end up activating their cell cycle, not the ones that return to a quiescent state, will end up fusing with the myofibers. It's like their final goal, because there is like an internal clock regulated by the activation of multiple genes that end up causing their fusion with the myofibers. So once they are activated, they will fuse even if there is less damage or not. And also because the IGF-1 factor is being released in the same way, in similar increases in both groups, even though there is less damage. So the group that has less damage is activating more satellite cells, and since this factor regulates fusion because it regulates differentiation, we know that we will have the same amount of fusions, or even a greater amount of fusions. I think that they will have a greater amount of fusions because of the 6th citation that proves they require a repeated bout effect to row. We will have a similar, or I would say greater amount of fusions, even though we have less damage. So since we have less zones or places in the myofiber in which there is damage, then where the satellite cells that were going to the damaged zones in the other group, where are they going now that the zones damage are not there, because this group is receiving less damage? Where are they going? Well, they are certainly not going to these incredible amount of micro injuries inside of the muscles, because there is no incredible amount of muscle micro injuries in this group, because they receive the effects of the repeated bout effect. So this high amount of muscle satellite cells are directly fusing with the muscle fibers, because the IGF-1, which we have already said regulates the aspect of satellite cell fusion, is being activated even though damage has decreased. So they have less places to fuse to, and they have the same amount of factors that regulate their fusion. So the answer to all of this is that they will fuse to zones with no damage, because they are less zones with damage, in this case, because this will precede the repeated bout effect, but effects. And there is the same amount of a promoter, right, of satellite cell fusion. So less places to bind to, and the same amount of promoters of binding, right? So they will, as a conclusion, as a logical conclusion, end up fusing with fibers that don't have damage. Also, we have already said that damaged fibers don't have anything in particular that is necessary for satellite cells to fuse. As a matter of fact, the fusion is regulated internally by satellite cells. Nothing. Well, the IGF-1 promotes fusion, right? But no factor, right, regulates the changes the structure of the satellite cells, because in order for them to fuse with the myofibers, because it is regulated internally by its genes. You can only promote the activation of that genes, but you cannot make it so that an external factor regulates the shape of the satellite cells so they can fuse. It is regulated by genes, which are regulated by other factors, but these factors are not directly regulating the muscle satellite cells fusion, right? They're indirectly regulating them, like IGF-1. It doesn't directly regulate their fusion, but it does so indirectly by activating some genetics, some genetic factors inside of the cell that lets it fuse, right? By differentiation of the cell. So it promotes fusion, but it's not the direct cause. The genes inside the cell are the direct cause. Okay? This is an indirect cause and still elicits fusion, even though it's if we have the causal chain, that let's say that IGF-1 promotes MYOD activation, which promotes differentiation. If we see this chain, the IGF-1 growth factor is not directly behind differentiation. So we cannot say that it's the direct cause, but it is in the causal chain. So it is involved in this effect, but not directly. The thing that is directly involved is the myofibers, which is how can I say it? An area inside of the nuclei of the cells of genes of how can I say it? Well, it's just a gene inside of the cells, right? These directly contributes to that. These indirectly contributes to that. Both are in the causal chain. Both end up causing this effect. But one is directly causing it, and one is indirectly causing it, right? Kind of a lengthy explanation, but that's okay.
Can you believe that shit? So, once satellite cells were identified as an autonomous cell population and subsequently named, their role in non-contractile-mediated physiological muscle regeneration following injury was quickly recognized. What was quickly recognized? That muscle regeneration following injury was quickly recognized. Oh my god, yeah.
First of all, regeneration happens only when the state of the myofibers has been completely compromised, which means that it happens when the myofiber was completely broken and it died. The cell itself died. So yes, damage can only be the cause of regeneration. And we are going to explore in a second core differentiation, the differences between regeneration and hypertrophy.
Now, here comes the big part. Unaccustomed activity of any type can be injured.
And it is saying that damage elicits some kind of growth inside your muscles, but it is not saying that it is the only thing that does so. And also, if it did say that, it would contradict the other things that it says, because as we have already seen, this article contradicts everything that you say. So it cannot agree with you, right?
What are they talking about there? Are they talking about people going to gyms lifting weights? What are they talking about there? They say unaccustomed activity of any type can be injured. And how do we regenerate muscles? How do we get more of these nuclei? How do we get more of this satellite cells? Through injuries, regeneration, injury.
Not necessarily. We have already proven all of the ways in which you can activate satellite cells and elicit myofibers growth without injury. He's just repeating the same thing over and over again.
So any activity can
That's why I don't watch every one of his videos.
literally injure the muscle fiber, and then what builds muscles? Protein synthesis must exceed that breakdown. So do you need a gym?
No, well, I've already criticized that in the other video, but you still require stimuli. And you're not even talking about what protein is you're referring to. It could perfectly be the case that you're talking about sarcoplasmic proteins, which help with ATP production, which is needed, funnily enough, for myofibers growth, because for you to produce more proteins, you require ATP. So since these sarcoplasmic proteins are involved in the production of ATP, which is involved in the production of in the process of getting the myofibers bigger, then sarcoplasmic hypertrophy is needed for myofibers growth. And that's another point, because in the other video I made another point about why sarcoplasmic hypertrophy is needed for myofibers growth, but that's something that again I will cover in the second core.
Do you need a gym to build more muscle cells? The density of the muscle cells? Do you need a gym to cause the...
You cannot build density of muscle cells. You can only... First of all, myofibers packing is not density of muscle cells. It's density of the sarcomer. The sarcomer is not muscle cells. Muscle cells are inside of the sarcomer. The muscle cells density is not changing. The thing that is changing is the density of the sarcomer. So that is wrong.
These injuries, well, because unaccustomed activity of any type they're talking about can be injuries. Even walking upstairs. If you're accustomed to walking up a set of stairs, a flight of 20 stairs you've never walked up before, you're accustomed to doing it, guess what it's going to be? Injuries to your muscles.
But going up in... sorry, he's contradicting himself again. If you go up a set of stairs, you are eliciting more concentric action than eccentric actions, because you elicit eccentric action of the muscles when you're going downstairs, like a study that they mention right here that proves that endurance exercises produce muscular hypertrophy, right? Have a study right here that proves that endurance exercise, funny enough, can produce myofibers growth. Funny, and it can activate muscle satellite cells, which he's going to blow his head off when he reads it.
Muscle fibers, that's right.
So he's contradicting himself because he's basically saying that concentric exercises are producing enough injuries to elicit satellite cell activation. So he's saying that concentric exercise is good for producing injuries, even though he only does eccentric exercise. Yes, concentric exercise produces injuries. That's why you should do full ROM exercises. Well, he's wrong again.
Especially if eccentric contractions are involved. Yeah, it's not the eccentric, it's always the eccentric. That's why
it's incredible because in each one of his videos, he either repeats his points that have already been debunked, or he's directly wrong.
Superior, because eccentric exercises cause damage, and you get more of these progenitor cells, so it must be superior. Okay, so especially if these contractions are involved. But muscle damage is specific.
No, sorry, what has he said just now?
And you get more of these progenitor cells, so it must be superior.
No, you don't only get more progenitor cells by damage, and your study mentioned, and I believe I have mentioned before, and it is in the video, this video, that mechanization expands the satellite cell pool, so it's not the superior way of training.
Okay, so especially if these contractions are involved. But muscle damage is specifically apparent with mechanical levity, used to induce hypertrophy. Oh my gosh, it's used to induce hypertrophy. Yeah, in both rodents and humans, so that must be hyperplasia. Must be hyperplasia.
Hyperplasia has nothing to do. First of all, hyperplasia hasn't been ever proven in humans, so that points fail by lack of evidence. And second of all, when more myofibers are being constructed in your muscles, the amount of myofibers that are being constructed by the fusion of myogenic precursor cells, like for example satellite cells, well, they differentiate into myoblasts, and they end up fusing into myotubes or T-tubes, right? This is something, again, for the other video, it's on the second core. But when these satellite cells fuse and form myotubes, they do so that they replace the myofibers that end up suffering necrosis and died. So they replace a whole entire myofiber that has died, a whole cell, because myofibers are cells that has died. So it is not producing more units or more myofibers inside of your muscles. There are no more myofibers or cells or muscular cells, which are not satellite cells. There are no muscular cells. There are no more muscular cells being produced inside of your muscle. These myoblasts are only replacing these myofibers that have died. They do not produce additional units.
Because if you get more cells, those cells hypertrophy. Oh, well, you have more, if you build more cells, it's already hypertrophy. Does that mean you can see it? No, under a microscope you could. But now you need to see that breakdown of protein to see that mass. The nature and magnitude of
not enough. But well, I have already explained why.
Damage.
Not enough to produce or elicit myofibers growth.
It's complex. It's extremely complex, so some people can't understand that though, because they're so dumb. They think Rojas building a muscle in his garage.
Yes, you are building muscle if you train in such a way that you have enough protein, which is something that we will talk about in the next video. Because he didn't even search or investigate more about the amount of protein that you require to elicit myofibers growth or to maximize a protein synthesis. He hasn't investigated a lot. So he says something like, which is completely stupid, that these studies that talk about 1 gram of protein per pound of body weight, they are only talking about people who are unaccustomed to exercise, which is completely stupid because they receive more damage. So in the case of these studies being based on people that receive more damage, which are newbies, they would have a bigger amount of proteins requirement for them to grow. So you cannot say that the amount of protein that they consume is lesser than the amount of protein that someone who is advanced will consume, because an advanced lifter damages his fibers less. Even you, who thinks that doesn't have the repeatable effects acting upon his fibers, because it is acting upon your fibers if you train weekly, since the repeated bolt effect has a duration after the last exercise of 8 to 12 weeks. So for your fibers to get unaccustomed to exercise again, you require 8 to 12 weeks, so you are not really buried lifting. And, well, if In summary, if these studies were about newies, you couldn't say that the protein that is being recommended in these studies is not enough for advanced people because newies consume more proteins since they receive more damage than advanced people.
All right, and may involve a degree of muscle fiber degeneration and necrosis. It does. There's a degeneration in muscle damage, and then it's followed with a regeneration. That's why I say you need to be in an anabolic growth state.
No, degeneration in muscle fibers does not require regeneration. The regeneration stage is a stage in which the entire fiber has died. You can still regenerate a little zone of the myofiber if there is a micro injury, which elicits growth, right? But it's not the only way to elicit growth, which is the thing that he has wrong.
So you don't get an apoptosis instead?
He gets wrong, sorry.
So you get a degeneration
Not a native English speaker.
and necrosis, and then you get a regeneration. That provokes satellite that provokes satellite cell activity. And what?
Yes, but it is not the way to provoke satellite cell activity. He still gets the same thing wrong over and over again. He repeats the same thing over and over again.
Regeneration. A recent example of extreme muscle damage with exercise, in humans was provided by Mackey and Kazier, where very high volumes of eccentric weightlifting elicited a modified regenerative response. However, very high volumes of eccentric weightlifting, they say eccentric weightlifting elicited a modified regenerative response. Yeah, causes injuries. So there's a regeneration now.
So now high volumes are right, because all of the studies that use high volumes usually can make their subjects train multiple times a week so they can maximize the weekly volume. So if you're pointing out to a study which has high volume and you're saying that it is positive for growth, then you're basically saying that a study in which there is no period lifting, but the modality that you call chronic lifting proves that you're right. Because muscle growth is being elicited on chronic lifters.
In this circumstance, total cell cells in muscle repair is initiative, since the reconstruction of large portions of muscle fibers or the formation of entirely new muscle fibers. Okay, so the reconstruction of large portions of muscle fibers would be minimal amount of damage that you're doing in the gym. But if you're doing
No, that's not minimal amount of damage. Minimal amount of damage are micro injuries, no, not large portions of muscle fibers. It's literally the opposite. When you say large portions, you're not referring to micro injuries, which are the ones that cause growth. Also, they are not the only thing that cause growth. Again, I just have to clarify that every time, because he could skip up to this point and think, well, he has said that damage elicits growth. Yes, but I'm not saying that it is the only thing that elicits growth. I already proved that. So, paying attention also elicits growth without damage.
Massive damage, like they say, high volume elicited weightlifting, elicited modified regenerative response. If you do massively more damage to the muscle fibers, guess what's going to happen? Or the formation of entirely new muscle fibers necessitates them. So not only are you going to get a reconstruction of a large portion of muscle fibers, you will get a formation of entirely new muscle fibers as well.
Yes, which is not hyperplasia, is the replacement of the fibers that have completely died. Replacing your dead myofibers is not the same as producing additional units of myofibers. As a matter of fact, all of these studies have only proven, even the study that explains regeneration versus hypertrophy, in muscle fibers, yes, have only showed that this regeneration response does not produce additional units of myofibers. It only produces the same amount of myofibers that have been broken. There is no study in everywhere on Earth that has proven that these kind of stimuli that completely destroy myofibers, which is the usual stimuli that newbies receive, can elicit the production of additional units. They are just talking about the regeneration process in which myotubes are being formed, and the myofibers that have completely died, the entire muscular cell that has died, is being replaced by a new muscle fiber formed by myotubes. It is not talking about myotubes forming a new fiber, a new myofiber, which is not replacing a previously dead one. A previous fiber that has died. They are always talking about these myotube fusing to form myofibers that replace another myofiber that has already died. They are never talking about these myotubes producing new myofibers that are not meant to replace the ones that died, but that are just extra myofibers or additional units. Nowhere, in no study, has that ever been proven or elicited in any way.
Well, depending on how much damage you're causing to the muscle fibers. Now, that's not going to happen if you're not eating an anabolic growth state. I'm sorry, it's not going to happen. Interestingly,
as you reply earlier, protein synthesis to be greater than protein breakdown. But that's a necessary antecedent, not a sufficient cause. Two different things. It is necessary to grow, but it is not sufficient to grow.
Because it got to go through a degeneration and a regeneration. Interesting, that's why you got to go over a week period to allow it to degenerate. The first 3 days or 2 days, there's a degeneration, and then the rest of the week following before you come back to hit them again.
Not necessarily, because the repeated bout effect lets you repair your muscle fibers faster. And also, monosatellite cell differentiation and fusion happened in a span of less than 20 hours. So your muscle fibers, there's a study right here. In the second core, you can see if you want. Like, where is it? You have the first division 2 hours in vitro. So these satellite cells, which will regenerate your muscle fibers, are already activated 12 hours after training. So what you're saying about 7 days is completely stupid. And I have the image right here about what he's talking about. You require, say, 7 days for maturation, but regeneration already happens 4 days after this training. So you don't require a full week, and you don't even require 4 days, because the amount of damage that you receive thanks to a repeated vault effect is not so great. So that the next time you train, you have all of the function of your muscles completely compromised, right? Because repeated vault effect protecting you, and it's also necessary to grow. And apart from that, the more you train, the faster this cycle is. So most people, if they train consistently, probably know that once they have been training for a few weeks, like 4 weeks or 5 weeks, the time that they take to regenerate from one training to the other to recover all of the function of their muscles. And this cannot be explained by sarcoplasmic hypertrophy, because sarcoplasmic hypertrophy does not elicit an considerable increase in muscle strength and strength, so that it is justified that from one day to another, in which you have completely broken your myofibers, in accordance to what he says, that from one day to another you are completely regenerated, or you recover all of your strength. This sarcoplasmic hypertrophy related strength should be so strong, should have an effect so strong, so that it replaces the strength that all of the myofibers, which I say are a lot, all of the myofibers produced before being broken or injured, right? And it's just such a stupid hypothesis that he also haven't proved in any way, shape, or form, but, well, he always does that. Warranted assumptions are his usual arguments. And, well, as I already mentioned, the repeated vault effect makes it so that you regenerate faster. And this can not also be explained by androgenic receptor gains, because these androgenic receptors are being incorporated in a very slow manner, because they are mainly controlled by genetics, as I have already explained in my previous video, using the article that he linked, which is quite funny. They are mainly controlled by genes, so even though you can change the amount of androgen receptors that you have in your muscles training, the change is incredibly small, so small that you cannot just justify these strength gains by using just the increase in androgen receptors, because in all studies they had to regulate androgen receptors by making knockout mice or modified mice that have different genes. So the main way to modify androgen receptors, or the amount of androgen receptors that you have in your muscles, is by changing your genes. So this exercise, this continuously exercising, cannot change the concentration or the amount of androgen receptors that you have in your muscles that much, so that you can explain the strength gains that you have received. Obviously, the strength gains that you receive are from the central nervous systems. No one of these factors contribute with this, and the central nervous system does not adapt in such a short windows of time. So, again, we only have one explanation for this. This regeneration stage is being moved some days before what is said here, because of the repeated vault effect. The repeated vault effect basically makes you stronger, less weak, and it makes you basically better in every way, so I don't understand. And also, it helps you with inflammation, and it reduces the amount of oxidative stress that you receive. It is very good for you. The repeated vault effect is something good, not something bad. It's very stupid what he's saying here.
It's going through a regeneration, you understand? All right, interestingly, it seems that cell expansion with mechanical loading often exceeds what is required to repair muscle damage. Coordinated regulation of proliferation, differentiation, and apoptosis in solid cells likely ensues. That solid cell proliferative overshoot does not result in access fusion to muscle fibers, while recent evidence provides insight into how solid cell exhaustion...
He's going to contradict himself with what he's going to say. But basically, when mechanical loading causes this cell activation in a very large scale, regulation of proliferation and differentiation by other factors like FGF2 and IGF-1, all of that, nitric oxide and calcium, and a lot of substances that are released by just mechanical tension, yes, and have been proven to be mechanistically linked with mechanical tension, which means that there is a cascade of effects that mechanical tension produced, in which damage is excluded, which end up eliciting the release of these factors, right? And, well, basically, all of these factors that are released by just mechanical tension regulate your growth. Or, well, the amount of satellite cells that are being activated and used.
Is prevented by exercise. So think about this. Your coach Blaha, he's exhausting the satellite cell exhaustion.
No, you have all of these mechanisms. It's literally right there. I can't believe that you have just read it and you're ignoring something that you have read seconds ago. You have to be incredibly blinded by your own bias in order to do these things, but well, it's nothing that we couldn't expect from this person right here.
You understand me?
So how much hours does this stream has right now? 1 hour. Not much.
But this regenerative stuff does not increase the satellite cell number. How do you increase the satellite cell number so you don't end up like Coach Blaha in Sarcopenia stuff? Does not increase the satellite cell exhaustion. Understand me? But this regenerative stuff does not increases the satellite cell number. How do you...
Yes, it does. And the study that you were talking about mentioned how an increase in mechanical loading elicited an increase in the amount of satellite cells that are in quiescent state in the near pool. Where is it? Well, let's look for mechanosensors. So, I believe that it was there. Mitral. And this is something that I also mentioned in my docs, which is quite funny, which is this interleukin factor that is related to just mechanical tension alone. Which is funny because this means that this article is kind of proving me right again. And right here, we have the thing that I was talking about. Satellite cells also increase in number in response to in vivo mechanotherapy. So by mechanical loading, the amount of satellite cells that you have in quiescence also increases. So this article right here has proven him wrong again. And I'm using just his article.
Increase the satellite cell number so you don't end up like Coach Blaha in Sarcopenia. It's really easy. Regenerate muscle fibers by injuring them.
Sarcopenia can only happen if you don't train. You cannot have sarcopenia if you train without doing excessive exercise. Excessive exercise would be something like 4 hours per day or something like that.
So that the progenitor cell, when it comes out, it can split, divide, and increase the number. But it won't split, divide, and increase the number if you're a chronic lifter in the gym. Get it, man?
Yes, it does. And we have proven in the last video that, and in this video, because he's saying, when he talks about chronic lifters, he's talking about people who trains in a frequency bigger than 1 in 1 week, right?
Yeah, he's not going to do that, okay? So under less strenuous conditions such as moderate...
We have all of the studies of House of Hypertrophy that talk about my freelance growth that prove him wrong.
Intensity resistance exercise. Damage induced by contractions is characterized by disruptions to the internal structure of muscle fibers that involve physical forces and/or excessive calcium release from the sarcopenia.
Is but calcium is also released by just mechanical tension, even if the structure of the cell has not been compromised, which is the reason why you can link mechanical tension alone with the release of all of these substances that regulate the satellite cells cycles, because nitric oxide, calcium, and all of that are being released even if the structure of the cells, of the muscle cells, has not been compromised. So if we know that the muscle cell received no damage, it's necessarily the case that the muscle tension signaled this release of substances. Because if it didn't make the cell release its substances by damaging it, it must have necessarily activated something that signaled this cell to release these compounds by its own. Or we have another explanation that is very easy, that comes from an increase in activation of motor units. When you activate more motor units, the amount of compounds that are being exchanged between the cells and the exterior is greater, and there is greater leakage. So there is a greater volume of nitric oxide going from one place to another, calcium going from one place to another, lactic acid going from one place to another, and that ends up being a regulator by a positive regulator of activation of satellite cells, because their concentration is being increased by just activity alone. It's not being released because of damage. We're taking the case of cells that had their structure not compromised, right? Just by activity alone, we have this leakage.
Plasmic dictum, as well as localized focal damage to the sarcopenia. In concert with sarcolemma, destruction factors released from muscle fibers and subsequent.
You can see how much he cares about reading the entire study instead of just reading the abstract. Here, he's reading everything at 4 times the speed. So he's not even understanding what he's reading. This again proves this right here, confirmation of bias. He only cares about the things that proved him right inside of this article,
Immune cell infiltration
ignoring all of the things that casually inside of this article prove him wrong.
are the likely main factors of satellite cell fusion to muscle fibers with traditional resistance exercise expanded on
You have also immune cell infiltration in the case of just mechanical load.
below. Well, they're standing there. Man, it's going to take forever to read all this through here. The study of mechanical load-induced adaptations and notice.
If it takes you that much to read a study which is far how can I say it? Which is far smaller than the ones I had to read for all of this thing that you have right here, that basically proves that you don't read much studies, because this is not the average length of a study. If it seems much for you, then you're basically proving that you don't read much articles. You only read the abstract of them.
Primarily induced story hypertrophy synergistic. I can't control this, man. It's going to take forever here. Solid cell proliferation, markedly and fusion to muscle fibers in response to synergistic adaptation, making it effective model studying satellite cells. Okay, let's just go on. I think that was the most important. Satellite cells.
Yes, it was the only thing that you try to take out of context to prove you wrong, and even so, it doesn't prove you right. Prove you completely wrong.
Accelerate muscle fiber reconstruction given their development after muscle membrane resealing. Yes, they do a lot. They build muscles, yes. Even after release of overload without satellite cells function, normalized fiber size is not compromised. If membrane damage is not catastrophic, so this is very complex, bro.
Not really complex. This is one of the articles that are easier to understand out of all of the ones I have read right here. You have some articles that are far more difficult to understand. For example, the one that talks about MRF in differentiation stages, muscle regeneration can be divided into several stages. You have myogenic regulatory factors, who have MYF5, BUC7, MYOD. All of these factors are, and all of these genes right here, or transcriptional factors, because, well, you don't even know what transcription is, but it's basically the process of creating proteins that acts as messengers or as structural units. Well, it's far more difficult to understand that what you're watching right now.
Excessively damaging protocols involving force intense running in the absence of self-fusion results in significant muscle impairments. This is what they tell you here. They say, conversely, excessively damaging protocols involving force intense running in the absence of satellite cell fusion results in significant muscle impairments. Well, that kind of sucks. That's their coach law. But that's pretty soon.
Sorry, but if you say that he's just producing adaptations that prevent damage from happening, then you can point out to blah. You are, again, contrary to yourself. And the protocol that you try to follow is excessively damaging yourself, because you don't have the protective effect of the repeated alt effect. So it's kind of funny that you read something that contradicts you, and you trying to twist it in some way so that it serves as evidence for what you say.
Coming to him with theory. Thus, the degree of positive damage during load-induced hypertrophy, as well as the specific hypertrophic stimulus, likely determines the necessity of satellite cells for successful hypertrophic adaptations. Yeah, they're really important, man. Without them, forget it. Yeah, I don't even know what this one is here. Fusion
even though that same study talk about increase in muscle myofibers size without an increase in muscle satellite cells.
Loading induced muscle hypertrophy. Niche derived Qs. They have a Q. Why not Qs? A challenge to understanding what media size fusion to add muscle fibers with loading. This is difficult to parse the effects of external Qs on muscle fibers and/or mononuclear cells versus the cell line effects of mechanical loading.
Which is the first thing that I explained about that article.
Satellite cells, okay. Muscles can grow appreciably without myonuclear excretion when hypertrophy is induced by genetic or pharmacological means in vivo without tension or damage. Yeah, I know that.
That directly contradicts what you say, because it's talking about growth in myofibers without an accretion of muscle satellite cells. It's really funny that he says something that completely contradicts himself, and he doesn't even notice.
That just means something's expanding, but that doesn't mean you're getting more cells.
No, you are getting more bigger myofibers. It's talking about myonuclear accretion, which means that it is talking about muscle fibers, talking about myofibers. It's not talking about the sarcoplasm nor the sarcolemma. It's not talking about the structure that holds the muscle fibers in which the sarcoplasm is. It's directly talking about muscle fibers. So you cannot blame it to sarcoplasmic hypertrophy because the sarcoplasm is outside of the myofibers. So it cannot be the thing that is causing them to grow. It's an external thing.
That just means something's expanding, but that doesn't mean you're getting more cells.
Wrong. Why? I've already explained why it's wrong.
This suggests that the mechanical loading is a key stimulus for cellular selectivity during growth. Yeah. Some studies report that cells on adult muscle fibers can be activated in response to stretch and shear stress. Well, they can't. Let's say I stay in a stretched position for an hour or 2 hours. When I do a lot of stretching, like in martial arts, a lot of that stretching is going to cause a break in the muscle fibers. It's going to cause it to tear.
No, it's just a byproduct. It's not the thing that it's causing them to activate. Cells that do not have their structure compromised, well, I have already explained like 30 times in this video, but there are cells that do not have their structure compromised and are still releasing chemical substances that elicit saturated cell activation. So you cannot blame it to the byproduct of muscle tension, which is not even something that is implied by muscle tension, because muscle damage is not proportional to muscle tension. Therefore, you cannot say that one thing causes the other if they can vary independently one of another. And, well, damage does not serve to explain what you're trying to explain. And it's not the thing that is eliciting, in the cases, the activation of the mechanism, considering that there is a mechanistical link with the activation of said thing with mechanical tension, right? Which means that we have a mechanism, step by step, that proves why muscle tension elicits myofibers growth, right? Which doesn't happen with the byproduct of muscle tension, which isn't necessary for any step of that mechanism that ends up activating which is involved in that mechanism that ends up activating the satellite cells. This damage is not necessary in any steps. Let's say that the mechanism is 4 steps in length, up to its final effect. Well, damage is not necessary for any of those steps. You can explain all of the mechanism by just the consequences of muscle tension abound. And muscle tension, well, since muscle tension does not cause damage, because you can avoid it because they are not proportional one to another, we have to rely to explain this mechanism on things that always accompany mechanical stress. So it's also obvious why damage wouldn't be in any of those steps that end up causing myofibers growth.
All right, and shear stress can cause it. I don't know what's stressing it, but something stressing the muscle fiber can cause it to damage to whatever. There's a response to activate.
That's a byproduct. You cannot blame the byproduct. You cannot blame the final effect on the byproduct, especially when it's not always present, and especially when it's not involved in any of the causal effects of the mechanism that links mechanical tension to myofibers aggression, right? Because obviously this mechanism to operate needs to include things that always accompany, or let's say that are caused by mechanical tension. So you cannot have damage inside of this mechanism, and you cannot explain it by damage. And not only because of this counterargument, but also because of all the other counterarguments I have explained inside of this video.
Suggesting they are mechanosensitive and can act self-autonomously with contractions in vivo. Mechanical strain on myogenic progenitors in vivo causes...
You have just read about mechanosensors, so you have structures that only they are specialized to sense mechanical tension inside of your muscles. And I have already explained it in this video, but these structures are strictly related to sensing tension. Their main focus is to sense tension. They do not care at all about the amount of damage that is being produced. So if you point out to mechanosensors, you're basically proving, again, yourself wrong. It's very dumb, this.
Sound like cell proliferation, but inhibits differentiation fusion into myotubes. That's not good. But these are in vivo, man. They're doing tests.
Mechanical strain on myogenic progenitors in vitro causes satellite cells proliferation but inhibits differentiation and fusion into myotubes. Why? Because, first of all, you receive mechanical strain. And you don't want these cells to directly differentiate and fuse after receiving this strain. You want them to first proliferate. This has to be an ordered mechanism.